Hi everybody, I'm Stephen Woski. I'm an associate professor at the University of Alabama in the Department of Chemistry and Biochemistry. Thank you for your interest in our graduate program and thank you for your interest in my research program. I'm looking forward to this opportunity to tell you a little bit about what me and my students have been working on over the last several years. So to get right into it, we are in the area of chemical biology. Primarily, we use tools of organic chemistry, synthetic tools, as well as spectroscopies of various sorts in order to address problems in biological systems. And in particular, we're interested in nucleic acids. So just to start with this idea, this is of course organic chemistry view of what DNA looks like in the center of the slide. You can see that you have the bases that have complementary hydrogen bonding patterns to a base on the opposing strand. The two strands go in opposite directions, and ultimately when you look at the three-dimensional structure of this is the right-handed double helix that you're all very aware of. What makes nucleic acids especially important are the fact that they are the key molecules for storing and transmitting information in biological systems. DNA is a molecule for long-term storage of information. This is where genetic information in most things is kept. And then, of course, in order to express this information, to do something with it, a working copy in RNA is made where it then interacts with tRNA and ribosomal RNA in order to make proteins which catalyze most of the reactions. So as a starting point, my group is very interested in how the structures of nucleic acids how they come to be, why are they the way that they are, and how does this affect the way that nucleic acids interact with other molecules, small molecules or proteins in a biological system. We are also interested in using some of the compounds that we make in order to utilize nucleic acids and their ability to carry information in order to make new probes, things that would tell you if a certain sequence, say from a pathogen, were present in a sample. So to kind of give you an idea of our approach, we've primarily focused on replacing the nucleobase. So this is a deoxyadenosine residue. You have this purine base, and each of the residues in a nucleic acid has one of 5 bases of guanine, adenosine, or cytosine in both RNA and DNA. And then, of course, DNA has thymine and RNA has uracil. But these bases are essentially 2 flavors. You have a bicyclic purine, which is flat, and then you also have a monocyclic pyrimidine, which is also flat. And our basic approach is we're interested in what's going on with the bases, because this is where the information is being stored, and it's also the source of the main stabilization for nucleic acid structures. DNAs are double helical largely because of the interactions between the stacked bases. So in order to understand what the bases are doing, we'd like to essentially mutate what the base looks like and how it behaves in order to understand the interactions that are involved. What's involved in making a base pair? Why are only hydrogen bonding base pairs known? Why can't you use other types of non-covalent interactions? What are the interactions that maintain helical structures or other three-dimensional structures of nucleic acids? So these are all questions that we can address by modifying, or at least begin to address by modifying the structure of the base. One approach is to generate things like aryl C nucleosides, where we take a carbon-based aromatic system and use that to replace the carbon-nitrogen bond and some number of other nitrogens, for instance, in a nucleobase. We also have been interested in trying to do this as a modular type of system. These carboxymids allow us a way to put a carboxylic acid group in this position and be able to make a large number of modified residues by forming an amide bond between an aryl amine or some other amine and that acid. This turns out to be a better way to very simply label nucleic acids. And then finally, we've done a fair amount of work in peptide nucleic acid where the backbone is now really different. It's now a polypeptide backbone using this 2-amino ethyl glycine, and the nucleobase hangs off of it. This forms complexes with DNAs and RNA like you would expect, but does not have a charged backbone. It has a neutral backbone. So just to give you some ideas of the kinds of things we can do with these molecules, one nice experiment that my group has been working on, and we're still working on interpreting all the data from this, was very simply asking the question, what does the structure of a single residue contribute to the stability of a large DNA helical complex? And so we asked sort of a physical organic question: what effect does the identity of this group at the X position have on the stabilizing effect of this residue as it stacks? Do electron withdrawing groups make this stack better, or electron donating, or what type of interactions between the stacked bases are important? What we initially seem to find from this is that magnitude of the dipole moment is something that is most important, and this is something that was a bit unexpected and something that we're chasing down by looking at large numbers of residues in this position. This is just to give you a little bit of an idea of some of the things that or some of the chemistry that we use to make these. We have 2 or 3 different methods that we use to make nucleosides, which can then be converted into a phosphoramidite and used on a DNA synthesizer, which is basically just a complicated pump that delivers reagents to a growing chain of DNA on a solid phase. And we can make oligonucleotides in any sequence we want, including having non-natural residues at any position that we want. And that's our basic approach. One of the types of experiments we do to look at this is to do what's called thermal denaturation experiment. We very simply take 2 strands, one's complementary to the other, and these contain a non-natural base pair in this case. And we can heat the duplex up and watch for the strands to separate. Turns out that the absorbance at 260 nanometers increases as you go from double helical to single-stranded DNA. And from this you can get an estimate of the stability, called the TM, the melting temperature, or you can also extract thermodynamic data. You can get delta H and delta S for the transition out of this curve, and then of course you can work back to free energies from that. This kind of gives you an idea of what you can do if you carry this all the way to its conclusion, and that would be make non-natural base pairs. So these are things that we've completely designed. Our inspiration for a lot of this is crystal engineering. So for example, halogens are often arrayed in solid phase so that the halogen atoms are in contact with each other, or in contact with an electronegative element. This would be a halogen-halogen interaction, or a so-called halogen bonding interaction. We can also just think of things as hydrophobic edges or even shaped edges. Here we have what's essentially a convex face that would be interacting with a concave face, and we would predict that this would be a shape-based base pair. Finally, we are interested in developing new bases that do functional things. So one thing we're very interested in are bases that have no base pairing specificity, a universal base. This is an example of one of those that we've made. What's interesting about this is that it can be used as a wild card in a probe molecule. It has no base pairing specificity for A, G, C, or T, and so it can be put at any position and will accept any partner in a duplex. Even potentially more useful are fluorescent bases that do the same thing, and here's one that, or a couple that we have designed. We have not made these in either DNA or RNA yet, but these are universal and they fluoresce, and in particular they increase their fluorescence when they light up when they form a double helical structure. Finally, we are also interested in how molecules interact with DNA. One project that we're working on in collaboration with John Vincent's group in the department is to look at chromium DNA interactions. Chromium is a carcinogen. Chromium 6 is a potent carcinogen when inhaled, and it's believed that it gets reduced to chromium 3, which can then interact with DNA. And we are using NMR spectroscopy, working toward X-ray crystallography, and doing other spectroscopic experiments to characterize what the structures of these lesions look like, and with the goal of understanding how chromium exhibits the effect that it does. So, having said that, I hope I've given you a little bit of a flavor of what we do in my group. Again, I want to thank you for your attention and let you know that you are certainly welcome to reach out if you want more information from me or from anybody that you've spoken to or seen the videos of. And hopefully we will see you in fall. So thank you and have a good day.